Thank you. So first, it's fantastic to come to Kingston. It was a nice drive. It was easy, and it's great to have people that we can collaborate with so close and sort of build the Canadian connection here. So today, my talk is basically going to be looking at liver endothelial cells, and we'll talk about a little bit about what those cells are and why I think they're really exciting. But jumping to the very end of the talk, the whole goal is to make a cell that can do a job. And what you're actually looking at is a mouse's liver, where all the brown cells are human cells, and they're engrafting that animal. They're taking over and replacing the function of the liver endothelium inside that animal, and they're there for the life of that animal in that model. You could think of that animal as a patient. You could think of it as a research system, but it gives us an exploration space that we can start to ask how those cells do the jobs that they do and how we can get them to do jobs that we want them to do. So it sort of starts off as this concept of blood vessels having functions. We know that blood vessels are involved in every one of the body's organs. It turns out there's a bunch of different cell types that are in every one of the organs of us. Endothelium, fibroblasts, macrophages, NK cells. You go around the organs, they all are there. What's really exciting, though, is in the endothelium, we've got really good data that says that in each one of the organs, specifically capillary beds, are different. They're doing different jobs. We don't really know why they're or how they're driving those jobs, but they're doing different ones in each one of the organs. Right now, I'm using my gas exchange capillaries quite aggressively. My blood-brain barrier is keeping everything out. My liver is scavenging all kinds of stuff that's flying by, and my skeletal muscles are being well fed. But what tells those endothelial cells to do those jobs? They're critical to the function of the organ, but we don't know what drives it. So the structural part that's really the backbone of this is as you go through each one of the organ sets, you have continuous capillary endothelium. Can I get that to go away? Yeah. There's rumors of it being recorded. Okay, so the structural capillary types are actually different between every one of the organs. We have continuous that you see in the brain, which now it won't let me click. No, now we're locked in here. Won't go out that way, won't go out that way. We are apparently stuck. Let's see if we can click the button. Okay, we'll try clicking. Okay, so we have in the brain, you have continuous endothelium. This has a thick basement membrane and no holes. As you head into skeletal muscle, it's still very thick basement membrane, no holes. And then when you get into fenestrated endothelium, that's where you see absorptive tissues. So you have to start transporting things. Secretory and excretory organs have these types. These are fenestrated. Then we head into big organs that need to have large objects. Think of a red blood cell, right? It has to head out of an intracellular gap, have an incomplete basement membrane so you can get that red blood cell out of the bone marrow and back into the spleen for degradation. What's really interesting is it really tells us these capillary functions are mediating the organ functions. They're tuned to what that organ needs. And the liver is no exception. Actually, the liver has clustered fenestration, so a lack of basement membrane, fenestrations that are clustered in something called a sieve plate. But again, we have no idea what drives this, right? So there's big questions in the endothelial heterogeneity field. What drives and sustains this heterogeneity? It's really important because as disease impacts us, whatever happens to happen, whether we have lung disease or pick your organ disease, those endothelial functions are the ones that are lost. The liver stops scavenging. The capillaries in the lung stop their transport of oxygen. So we need to figure out how to get those functions back. If we can control the functions, we can repair them in theory with drugs. But in some cases, in monogenic disorders or acquired genetic disorders, we may be able to replace the cells because they are obviously not going to be able to gain back a function that is genetically missing. This is basically the research themes of the lab. What drives function, and let's get it back in interesting strategies. So I mentioned that we're interested in the liver. You can see a nice liver picture here. The reason why we're interested in the liver is it affects a lot of different people. 1 in 4 Canadians are actually affected by liver disease. There's hundreds of liver diseases, but if we wanted to study each one of them, that's an individual model. Luckily, the liver vasculature loses function in almost every one of those diseases. It doesn't matter whether it was drug-induced, metabolism-induced, cancer, or genetics, something like hemophilia, the liver endothelium is losing its functions. So let's look at the liver so you guys can have a bit of a roadmap and follow along as to what we're trying to do and trying to understand. So within the liver, we have a repeating hexagonal unit known as a lobule. Radially patterned around these lobules is something called the sinusoid. This is a nice linear unit. It's a simple way of understanding the liver vasculature. Here we have the portal vein delivering nutrient-rich and hormone-rich blood from the intestines, mixing with oxygenated blood from the hepatic artery. It percolates across the sinusoidal space and then heads back out towards the central vein and out towards the heart. Now, the cell type that we're interested in are the liver sinusoidal endothelial cells. This is the major capillary of the liver. And you can see many other different cell types that are resolving in that space. There's about 20 different cell types. They're all interacting and doing all kinds of interesting cell biology processes, but together they get all the functions. The nice thing is when you have the different cell types, they're in different environments. A oxygenated zone and a deoxygenated zone, which will be important later. So these LSECs have a bunch of really key functions that are in this space. They are fenestrated and they pass blood components across to hepatocytes. This is things where LDL is transporting through. They're scavenging biomolecules like bacterial fragments, tissue debris, and immune complexes. They secrete factors, many different proteins, one of which is actually factor VIII, which is missing in hemophilia A. And they stimulate hepatic regeneration. If you were to cut out 2/3 of somebody's liver, it'll grow back in about 3 weeks. It's a really amazing process, and LSECs are really important in that process. So the markers for these LSECs are CD31, a general endothelial marker, a couple of scavenging receptors of CD32 and LIVE1, and factor VIII as a secreted factor. Now, I mentioned that liver diseases are a problem, and they do affect many liver functions, whether it's drugs, genes, cancer, or metabolism. We're losing all these functions, right? So it's really a commonality that LSEC functions are lost to liver disease, but it also reminds us that we really need to study it. And unfortunately, cell culture fails. In cell culture, when you take those LSECs out of an animal of any organism, put them into a dish, they lose all their functions in about 72 hours. That's a real problem. Now we can't study these functions no matter what we cared about. They don't have them anymore. They go into a nice growth phase, they start proliferating, and they have none of these capabilities. So my postdoc, I realized that we need to make them. We need to have access to these cells if we want to understand them. We need them every day, and we need the exact same cell. It can't be different ones from different genetic backgrounds, all the humans in the room. That wouldn't work for us. So we turned to human pluripotent stem cells, a cell type that can become any cell type in the body. And just following this nice simple arrow, we just have to make the one we want, right? Now, this is clearly simplified for important reasons, but to do this conversion of a human pluripotent stem cell to an LSEC, the pathway, there really only is one that I believe in, and that's follow nature. Nature knows it worked for all of us. It's going to work again. We need to follow both development and anatomy, and that will give us a roadmap in making the cell and the cues that drive the maturation. So I'm going to skip through a ton of background and just show you what a human embryo looks like at about 30 days of development. You guys haven't seen this. It's well worth looking at. It's the 3D Embryo Atlas. It's a rather old paper, but it allows you to 3D spin models of actual human embryos. If we peel back the skin and the nervous system, we can look inside and see a gastrointestinal tract with a nice brown liver here behind the heart and vasculature in purple and blue and red. And when we zoom in, the intrahepatic vasculature is directly connected to two venous structures. One, the sinus venosus, and the second one's the veins. There's a lot of developmental biology, but it perfectly matches the mice and the zebrafish. What they can tell us is if you want to make these cells, the intrahepatic vasculature, it comes from these two venous sources. Simplifying it even more, we just need to make a vein. Luckily, the mouse biologists are really ahead of the game on this. They've been looking at this for many, many years. Vasculogenesis is a long-studied process in mice. So what we need is a way to follow this process through in mice. We first take the embryo, go through gastrulation to make mesoderm, and then the early mesodermal progenitors will then go through a vascularization process where the arterial angioblast, that's the firstborn endothelial cells, our endothelial progenitors of the vasculature, form. And then we have the venous angioblasts that form. They form the early vessels. The heart start beating. You get your first flow. Note, we haven't talked about organs yet. Then after you have these early vessels, the organotypic vasculature takes over. This is when the organ calls to the vessels. Angiogenesis takes over, and now you've taken a nondescript vessel and turned it into an organ-specific vessel, okay? Luckily, it's a good roadmap. We just followed it. Nothing special. With human pluripotent stem cells, one of the first things we have to do, especially good for mesoderm development, is take your cells and form them into something called an embryoid body. This is just a mass of cells. We pattern them through primitive streak and mesoderm with basic FGF, BMP4 and a Wnt agonist to drive our mesoderm population. Then we pattern it towards either an artery or vein faded angioblast with VEGF and Notch signaling. Luckily, our angioblasts all express CD34, so we can purify them. This turns out to be a really valuable process because we can purify the angioblasts, freeze them, and thaw them whenever we want. Then we can thaw out the cells, grow them in monolayers to get endothelial, either artery or vein, and then see which pattern's better in an organotypic-like environment. So the first step, just like in the embryo, is really getting towards an artery and venous faded angioblast. This process is relatively efficient now. We can generate KDR and CD56 positive mesoderm. In case you haven't seen these before, these are flow cytometry plots. Every dot is a cell, and these ones are expressing the two proteins. These mesodermal cells can then be patterned with high VEGF to give us arterial cells. Low VEGF gives us our venous population. Once purified, we can see that they're expressing all kinds of useful markers, like the arterial cells have CD184, the venous cells do not, and that's because of the master regulator transcription factors that drive the process. Long story short, it matches mouse development. As you start culturing these cells out, they do not forget from whence they came. These endothelial cells, for instance, in the venous side, as you grow them out, they maintain that high level of CoopTF2 where the arterial cells do not. And even at these early time points, remember this is before an organ, they're already responding to cues. We're able to take these cells and load them into a collagen tube. And we're able to see that in response to TNF alpha, very basic stimulus, we can upregulate ICAM, an inflammation-like marker. But it tells you that the cells are ready to experience their environment. They are responding. So let's give them something more fun to experience. We had to sort of look back at the liver sinusoid because we're trying to make liver sinusoidal cells, and we had to guess what the signals might be here. Remember, there's 20 cell types, extracellular matrix, physical forces. There's a long list, right? So we decided to go with hypoxia as it's a nicely gradated signal, high in the portal end, low in the central vein end. We've got basic FGS to keep everybody happy, TGF beta to decrease the amount of apoptosis in the system, and cyclic AMP mimics a whole bunch of signaling molecules like hormones and a bunch of ECM signals. So when we take our arterial cells, you can see them here, or our venous endothelial cells and put them under a hypoxic environment with those factors, what we're looking for is to see if they can upregulate LSEC markers, CD32 and LIVE1. Now, the arterial cells do actually upregulate it a little bit, but the venous cells upregulate it a lot more and a lot quicker. Looking over many conditions and many different timing strategies, we can actually see that in the filled bars, hypoxia, we get much more induction of these LSEC-like markers compared to the normoxia levels, and the venous population responds much quicker. They also upregulate a ton of other markers, actually including stabilin-2 and a bunch of other scavenging markers. If we link the concepts together, what this is showing us is that anatomy is important. Hypoxia is critical to drive it, but also developmental drivers are important as well. Veins respond significantly faster and more strongly than the arterial cells, although eventually development wants to catch up, and the arterials can catch up. So the markers that we chose are scavengers. One of the really important functional assays about LSECs is they should be able to scavenge biomolecules that are circulating around. One LSEC-specific ligand is called formalin-fixed serum albumin. You don't really need to know what that is, but it's something that should be binding to the specific scavenger receptor of stabilin-2. The FSA uptake is significantly higher when you have the scavenger receptors. Completely makes sense. You need the receptor to bind it. So that was really exciting. And then we looked for all the other functions. And that was a lot of work. They don't have any of them. They are not fenestrated. They do not express factor VIII. So what does this tell us? We chose the wrong factors, right? The signals that we put into that well were not good enough. So what's the next factor? So about 2 years after this question was started, because we did try a whole bunch of other things, we realized we have no idea. It's really hard to guess what that factor is because we haven't identified it here. We picked the one that was in all the reviews and literature, right? We chose that factor for obvious reasons. But what is the factor that makes the cells mature? Still don't know, but we can cheat. There's something that has all those factors. So we shifted the studies dramatically away from in vitro and went in vivo because that liver has all those driving factors. Now, our human cells are going to be rejected by any mouse that is normal. So we had to switch to immunocompromised mice that have no functional T, B, or NK cells. Also, their macrophages are a little bit messed up, but importantly, they don't reject our human cells. Now we come in with RFP tagged human embryonic stem cells. So our venous angioblasts are red. We had to make space in this animal's liver. We use a conditioning regimen called monocrotaline. It's a toxic alkaloid that causes endothelial damage in the mouse's endothelium of its liver. And then we can give a select advantage to our human cells, and we transplant them in the spleen using the vasculature and the normal anatomy to distribute our cells throughout the animal's liver. Over the course of the next 4 to 16 weeks, the cells grow and engraft and theoretically mature. And then because they're color-coded, we can purify them out with facts and isolate them and ask questions of what cell type did we make. Now, the engraftment was actually pretty good and quite stable for many days, and we were able to see that our venous cells were actually the best engrafters. Common question is, what about the arterial cells, the LSEC-like cells? Long story short, less mature cells engraft better. More mature cells actually go through bad processes and make really bad and weird-looking grafts. We don't like them. This is by far the best population to engraft at the time. The graft sizes in the non-parenchymal fractions, so lacking hepatocytes, are nice and big. We've got a big human fraction here. We've got lots of endothelium that's CD31 positive, and most of those cells are double positive for LSEC markers. Using factor VIII as an example gene, we can go about 200-fold higher than we ever saw in vitro, with levels that are very, very close to primary human LSECs that have been uncultured, directly sorted from a healthy human liver. So this tells us that whatever the signals are in that liver, they're there, they're functional, and they're doing the job. But one gene is obviously not enough. So as one usually does, when you want an unbiased question, you want to see what all the transcriptomic expression patterns are for these cells. In our system, it's actually quite easy because we can recover them and they're color-coded, so we can get them back for single-cell RNA-seq, which is what we did. About 6,000 cells were profiled. And what we can actually see looking at a wide variety of different transcripts, just pop them up there for people that are interested, is we actually have a big cluster, this green one here, that is pericentral LSECs, so the ones down towards the central vein. Periportal LSECs are actually this population here, and there's two different types of a general endothelium and a small microcapillary other cell type. We do make some fibroblasts, but those actually we were able to get rid of in a different study, and there's a very vanishingly small amount of immune cells. So most of the cells are endothelial. And if you look through the different expression patterns, you can see that we have a large expression of CD31, a bunch of different scavenger receptors. We actually do not express von Willebrand factor, but we do express high amounts of factor VIII and a bunch of the transcriptional regulators and other scavengers in the system. So the best part of this kind of data is you can compare to other data sets, which would be considered a gold standard. In this case, we collaborated with Sonia McParland at the University of Toronto, who had generated single-cell RNA-seq from 5 actually healthy human livers. So these livers, the rest of the liver walks around Toronto and is transplanted. So we know that these cells are not from a hepatocellular carcinoma or one of these tissue adjacents. So we could really compare and see if it was as normal as we could consider normal. The Harmony batch correction algorithm allowed us to merge all of this data together, and it taught us a couple of really simple things. One, we don't make hepatocytes. Okay, we weren't trying to. We also really don't make immune cells. The one that we do see is a couple of macrophages and a couple of T cells, but the vast bulk population is this endothelial fraction. Our cells directly overlap with pericentral LSECs and periportal LSECs with actually relatively little general endothelium. This tells us that we're really humanizing, or we have populations from central vein, pericentral, and periportal populations, but we actually don't engraft the portal vein. So if they have the right genes, do they do the right thing? That's why we're really here. I'm interested in functions. All LSECs should be able to transport blood. Histology showed most red blood cells in these humanized liver samples. We also saw in our red humanized samples, in this case, a human-specific Clav1 antibody, that the most macrophages and the most hepatocytes are in their normal natural residence. Remember, these mice are actually able to grow and live for a very long time, so we don't think their liver functions are compromised. They seem as normal as a mouse looks. We're really interested in factor VIII, though, so we looked a little bit more carefully. Our humanized mice have human-specific factor VIII immunoreactivity around the human nuclei and the cytoplasm of those adjacent cells. But more importantly, factor VIII should be secreted if it's going to work. So we use the human-specific factor VIII ELISA. Our non-transplanted mice do not have any immunoreactivity in the ELISA, but over the course of time, our adult transplanted model, we're able to see significant increases in the amount of factor VIII circulating in these animals. This is sort of a chart of hemophilia disease severity as it correlates to this plot. And if you happen to be a patient with very severe hemophilia, you would have less than 1%. Okay. Circulating factor VIII levels. As you get more factor VIII, you get into the moderate category for disease phenotypes, and the mild goes from 5 to 50, which we have. So, in theory, the levels that we're producing are in the mild therapeutic correction range. Is it completely cured? No. Also, this is just a mouse. Really careful caveats. But we're really excited because this suggests that these cells might have a therapeutic value that I'll get to in a few slides. So one of the other things that LSECs need to have is this functional fenestration capability. Remember that we can get our cells back, so this wasn't too challenging for us. We purified the multi-marker positive cells and did scanning electron microscopy, and you're seeing, looks like 2.5 cells there. When we zoom in, we can see these special functional fenestrated sieve plates. This is both an organ specific, so only the liver has this shape, and it's actually species specific. Humans, they're pointy, and rodents, they're round. But when we look inside the sieve plates, we see these 100 nanometer holes. Now, circumstantial evidence, this is the right size for a 20 nanometer LDL particle to go through to its neighboring hepatocyte. Now, LSECs are supposed to be involved in scavenging, and individual cells express lots of the scavenger receptors. You can see them all there. And when we purify multi-marker positive LSECs, they're able to really nicely bind to coli bioparticles. Through an opsonization process. They also functionally bind acetylated LDL and formalin-fixed serum albumin. So all of the functions that we were sort of expecting, we can test for in this animal model. So to summarize this sort of work, we were able to walk. It took a little bit of time, but we were able to walk from pluripotent cells through early developmental biology to specify either an artery or a vein. We found that that specification was important because the venous cells were able to respond significantly better to a minimal set of induction environment. Once transplanted, the functions were able to go up significantly, actually. That's where we got routine engraftment, and that's where we saw that the single-cell RNA-seq was actually matching the primary human LSEGs. Now, I really only care about functions. I'm really interested in the genes that regulate functions. And this system really saw a lot of those important functions, whether it's transport, simple exchange, but the specialization for the organ of fenestration, scavenging, and factor VIII secretion means that now, finally, we have a model to try to understand what regulates this. Now, at the start of the talk, I said this was going to be a cell therapy or a design towards discovery. This is the discovery engine. Now we can take pluripotent cells, not go to gene of interest, and see which one of the functions is lost. A direct link model, genotype to phenotype correlations. Also, we can make models where we lack a gene, and we can see if the disease makes it worse, makes it better, or a therapy can bypass a missing monogenic disorder. This is a powerful model, we believe, and we're in the process of exploring that next. So that was the one side of the talk. The other side was cell therapy. So returning here to factor VIII secretion. It's a really interesting concept that patients with hemophilia do not have factor VIII secretion from a variety of different systems, but most commonly, the 1 in 5,000 males that have this condition will be due to a factor VIII genetic disruption, most often a gene inversion. There are some factor VIII inhibiting antibodies, but if you're missing a factor, give it back. It's a simple therapeutic strategy, right? So factor VIII infusion is wonderful. It has been life-saving. Historically, it had some constraints that we won't talk about too much today because we've moved on to prophylaxis with recombinant human factor VIII that can be injected IV every couple of days. And now we have even longer half-life variants. This is great. We're into the weeks category for one of the variants now. There's also a bispecific antibody system that can bypass factor VIII. So this allows for non-IV injection, and there's a bunch of anticoagulation inhibitors that can also be part of the category as well. Factor VIII gene therapy is actually quite impressive, not just because I'm standing here where a lot of these things have been discovered, but there's a variety of ways of giving back factor VIII, the missing gene element, whether it's viruses to patients, viruses to cells, lentiviruses, or much more commonly and in clinical application is the AAV system. One of the challenges is, unfortunately, these vectors are generally episomal, especially for the AAVs, and over time we've seen loss of durability of the expression of factor VIII. Now, those trials are continuing, and better and new technologies will and are being developed. But what about this as a different technology? A cell to replace the source. Factor VIII producing cell therapy. There was one clinical trial from Sigalon that actually generated almost a random cell type that was producing factor VIII, and the body really, really hated it for a variety of reasons. So we are proposing to use LSEX to make factor VIII, really only returning nature to nature. The normal cell type that makes factor VIII is an LSEX. We believe that to be the best strategy. This is actually based on a long history of liver transplantation work in hemophilia. It turns out when a normal donor liver is given to a patient with hemophilia, their bleeding is corrected. So we know that the organ is sufficient to provide hemostasis. In very rare cases, the opposite has occurred, where a hemophilia donor was the source of the organ, so they can't produce any factor VIII from that liver. When it was transplanted into a normal recipient, quite remarkably, they did not get hemophilia. This tells us that there's extrahepatic sources of factor VIII, and through a long list of a different series of studies, different organ transplants, and in some cases cellular transplants, we actually know that factor VIII can come from a wide variety of spots outside the liver. But if you look around in the body's organs, the liver is massive. It's enormous, produces huge amounts. And this simple argument of one organ being completely corrective or very close to completely corrective suggests that this might be a good candidate organ to base our cell therapy on. Also, bluntly, it's where our system actually works. So that's part of it. So where does factor VIII actually come from in the liver? Now, I've already told you this, but for many years, actually, it was a question. Basically, in 2014, the world came to a consensus that it comes from the liver, and it comes very specifically from the endothelial cells of the liver. So if we want to build a cell therapy, we're going to build these endothelial cells, as I just mentioned. The goal is 1 dose per patient, and the goal is a lifelong, self-renewing therapeutic. But now we have new challenges. Now we need a high-yield, scalable production. Now we need high engraftment efficiency. Now we need therapeutic bleeding correction. Now we're suddenly in cell manufacturing. And that's where the challenges started, about midway through my postdoc. I realized that my protocol at the time was too labor intensive, had too many processing steps, wasn't scalable. I could make enough cells for a couple of mice, but this was never going to get to eventually the dogs that needed to be treated or the patients, the pediatric patients. Our estimate for a pediatric patient is somewhere around 800 million cells. So if you think of the scale of that, you need to have a lot of cells being produced. So we thought now's the time to change, and we changed everything. We changed how we specify mesoderm. We changed how we specify endothelial cells. We're still making endothelial cells that are venous, but it's a completely different mesoderm population. In this case, we have a CD235A positive cells. I'll get to that in just a little bit, but this is a special marker of a yolk sac hematopoietic developmental cascade. And these cells are still purified by CD34, so we still make venous cells. Yields do matter when you're generating these things. The old protocol is in blue, where we go from 10 embryonic stem cells to 1 cell that could be transplanted. That is just not good enough. Our new protocol goes for a nice proliferation phase in the middle, and we get approximately 1-to-1. An 11-fold yield, the graduate students, the postdocs in the audience, is 11 times fewer differentiations. So if you think it takes 13 days to make cells, now you only have to do it once a year to make cells on the same scale. Okay, so now we have new cells. Let's go back to the old model. So we return to our monoclonally treated mouse, our NSG mouse, with a new cell population asking a lot about factor VIII secretion. First, our cells do engraft. We get about 60% of the non-parenchymal fraction is humanized. What that really means is it's a completely humanized animal. I'll show you a picture in a sec. But they're generally endothelial cell at a very high frequency, and they still have CD32 and live1. To bring back the slide at the start of the talk, you can see that there's lots and lots of engraftment in these animals. It turns out it's actually 95% humanization. All of the liver vasculature is humanized, except for, and I'll outline it here, that. That's the portal vein side. It's the only cells that we don't seem to engraft, but basically all the other endothelium of that liver has been humanized. There's about 20 million cells in a mouse's liver, and we went to at least 8 months, and that's where we just ended the study. The cells are still very present and at the exact same levels. So now, can they actually make factor VIII? Simple analysis. We were able to see that the immunoactivity for factor VIII is right beside our human cells. And more importantly, our factor VIII ELISA says that we're getting about 15%. We've got nice stable big grafts engrafted into the animals. So we basically have 2 populations of cells that look to be basically the same. They're producing about the same size grafts. They're producing eventually, and they're producing about the same amount of factor VIII from that graft. So which one are you going to choose? What's the better cell therapy? The kind of lazy scientists said, well, the yield is better. Can we please go with the easier to produce factor therapy or cell therapy? But that's not really good enough. So we developed a competitive repopulation assay. If anybody's in the hematopoiesis space, we actually modeled it on that, where they can take CD45.1 and CD45.2 cells, mix them 50-50, and see who engrafts the bone marrow better. We're basically doing the same thing, but our cells are color-coded. So a 50-50 mixture of venous endothelial cells that are red or green when transplanted in the animal. If these two cells compete equivalently with each other, you get a 50-50 mix. If red is dominating, then it was a better cell. If green is dominating, it was a better cell. We tested a wide range of the different engraftment ratios, but it matched cells, 99% red all the way to 99% green. You can see all the different mixtures, but the really kind of boring output is what you put in is what you get out. These are matched cells. It makes sense. It's really important that this is how it worked out. But the nice thing is we can still read out very, very, very tiny graphs. So it doesn't matter whether you've actually perfectly matched your mixture. That said, still use 50-50 because it gives you a better resolution capability. So with the assay in hand, does it actually work? So we took our old protocol in red, our new protocol in green. So new protocol, old protocol. 50 days or 45 days later, it's very, very green. The new protocol turns out to engraft really, really, really well. We do not necessarily understand why, but I'll try to explain our guess. But the new protocol is 50-fold better at engrafting. The old protocol cells really, really suffer this process. Remember that when we put them in on their own, they're okay. They've got nothing to compete with. They're better than the mouse. But when we put a mixture of two cell populations that can fight for that same space, the one that wins is the new protocol. It doesn't change what engrafts. There's still purple cells, live 1 positive, 32 positive. They're still LSEX, and honestly, they also make factor 8. We checked in a couple other studies, but the challenging question is what makes them better. This type of assay, just like in hematopoiesis, can't distinguish that. It can't tell you what the problem is or what is good. What it can only say is this one is better than this one. It's a binary assay to model. So we have to start asking some binary questions. If we look back at our original protocol, as I mentioned, at the time, and in bad experimental design, we changed all the factors simultaneously and made a new protocol. But what we did do is modify two sort of stages, mesoderm specification and endothelial specification compared to our original protocol. When we looked at mesoderm specification, it turns out that the old protocol does not express CD235, the new protocol does. This is a hint. There is molecular differences at this very early stage. When we take these two mesoderm populations and put them through a very common venous endothelial specification, so the only difference is mesoderm, we can make our mixture of venous endothelial cells with only distinguishing feature being the mesoderm specification process. A 50-50 mixture results in a big green bias. So what we know now from this is that the mesoderm specification is driving this engraftment difference. The next step was taking the venous endothelial, our best mesoderm here, and putting it with two different types of endothelial specification. I'm going to skip all the ways that we did that because there's no difference. Once you have a good mesoderm, and the tag is that mesoderm matters, once you have a good mesoderm, the rest sort of comes out in the wash. So we have spent the lab now focuses on getting the right mesoderm and less about getting the right vascular cells, because that's what seems to drive this engraftment phenotype. Okay, so the next, and getting towards actually the real meat of this talk, the last little bit here, is that now we have the right cells. We spent a lot of time building a cell model and a very good amount of time developing an improved protocol, but now we have cryopreservable venous endothelial cells that engraft very high efficiencies in animals. What can they do to a really special mouse model? This is an immunocompromised hemophilia mouse. This is a very close collaboration with Antonia Felenzi. Actually, collaboration is a weak choice of words. We shipped her cells and she did all the work in Northern Italy during COVID. So collaboration doesn't even really hold a note to it. But what we were able to do is test our concept of a cell therapy because we could freeze the cells like they would go into a patient, pop them in the freezer, ship them off to Italy, and then our patients are ready to go with a surgical team. In this case, these patients, these mice, bleed severely and have severe hemophilia. They were treated with monocrotaline just like our mouse model, and the question was, did these cells engraft into the mice? Because the Flensi lab are actually pretty good surgeons, they were able to directly compare intrasplenic injection and intraportal injection on our behalf. Intraportal is something significantly more clinical. When we eventually get to a patient, this is going to be what's happening. We probably won't be delivering them in the spleen, even though it's easier for a caffeinated surgeon like myself to put my needle in the spleen. Engrafted into these animals, about 12 weeks or 16 weeks later, we actually found really nice big grafts of human endothelial cells that were expressing mature markers, LIBE1, CD32B, and stabilin-2. What was important here is that the engraftment nor the maturation was blocked by the hemophilia modeled environment. It was an important question because if the cells just didn't engraft just because they didn't have factor VIII, it would be unexpected, but it would have arrested the research. And it didn't work that way. So now, did they make actually factor VIII? Yes, I wouldn't be here talking if they did not. So it turns out that they do have immunoreactivity for factor VIII. It's the same versus intrasplenic or intraportal, just because the cells are there. We have nice red factor VIII being produced. So does this factor VIII work? Now, in this audience, people are more interested in this than probably the rest of the slides, but the Flensi lab were able to do a activation partial thromboplastin time, which allows us to read out factor VIII activity in the blood and track it over the course of the graft, which is kind of valuable. It's directly compared to something called Kovaltry, which is a recombinant human factor VIII, and it gives us some idea on how good the factor VIII being produced by our cells is. What you can see is our non-transplanted animals do not have reactivity or factor VIII activity, where both of our transplant groups are basically right on top of each other all the way through. So it doesn't matter how you deliver the cells, but we see a nice growth of the factor VIII bioactivity, and it stabilizes at 16 weeks. That was the end of our study timeline. We probably could and should have gone longer, but at that point, we were honestly had a second wave of COVID hitting Northern Italy, so it was time to end the studies. At that time, we took blood samples and found that the factor VIII levels in this hemophilia mouse model hit the magic 15% that we had seen in all of our other studies. So it really doesn't seem to matter what's happening, whether it's in a normal immunocompromised mouse or this hemophilia environment, we still get to 15%. Now, what should 15% be doing to coagulation? It should be shifting it to a mild bleeding phenotype. So we did a tail clip bleeding challenge in this mouse model. And we were able to see that the severe bleeding phenotype of the hemophilia mice, the NHG mice, but in all the mice that received cells, whether it was intrapleural or intrasplenic, they have a significant reduction in blood loss. And what was really important here, at least in my opinion, is that there is more space to go until we get a wild type level. This was mild, right? We went from severe to mild, not severe to cure. And I like that concept because wonderfully, all of this can be replaced by this. This factor VIII ELISA really gets us to a functional level rather quickly. Now, going forward, we'll need much more data than that, but it allows us to screen possibilities. Okay, so the summary for the second half of this talk is that when we go through to the developmental process, we actually have some interesting insights that BMP4 and activin do regulate a specific type of mesoderm. This mesoderm is the same type of mesoderm that gives you the primitive hematopoietic wave. It also gives you ventricular cardiomyocytes, not atrial cardiomyocytes. It's a really conserved developmental pathway. I didn't talk about it too much here. Be happy to answer questions if you guys have them, but this is something that is true for what seems to be all of mesoderm. So if anybody's working in mesoderm, it matters the type you make. And it doesn't just matter for fun. It turns out that this made our lives a lot easier for efficiency and scalability. We got more cells when we listened to developmental biology. What we didn't expect was that it was going to matter for engraftment. It turns out that this engraftment process is much, much, much better. That was an unexpected win, but the cells that we're producing are essentially the same. We really don't see differences between the protocols. Once the cells are in, they're doing the same job. They make factor VIII, it's bioactive, and it corrects the severe version of the bleeding phenotype, at least in the mouse model. So conceptually, does early development matter? Yes, it does. And it does actually control cell therapy manufacturing and efficacy. So with that said, we've got a concept of a cell therapy brewing, right? But could we give these cells to anybody in the room? No, they would fail for 1,000 reasons, not just the least of which is they express GFP. Useless, right? So let's move towards that. The last side of the talk is actually just a future concept direction towards a universal cell therapy. You guys all look diverse. You have diverse genetics. That means that we have to do some a lot of things to change these cells. We do have a protocol that we think that will work, that will be scalable all the way up to humans. We actually don't think that's going to be a major challenge. But if we want to get to the idea of a cell therapy with the properties of one dose per pediatric patient, probably in a 1-year-old boy, a self-renewing therapeutic that will grow with that liver as it engrafts or as that patient gets bigger, lifelong durable function. We can't have any immunosuppression because you don't want to give that to a pediatric patient. And unequivocally, I know it's only 4 letters, but it must be safe. This is something that's going to go in a kid for 80 years or something like this. These are cells that cannot lose function just because somebody had a hamburger or some Tylenol. That's unacceptable. So we have to test that too. So the first thing that is absolutely must be changed is monocrotaline. This is a DNA alkaloid agent. It causes DNA damage. This is not acceptable in a pediatric patient. We have already seen that we can move from the spleen to the portal vein, so that part is relatively simple and trivial, but dropping this is not quite as easy. Current theories are, and we have not tested them yet, is ischemia reperfusion injury. Pediatric liver transplants happen all the time. Thankfully, not all the time, but when they do happen, the ischemia reperfusion injury does cause endothelial damage, and the liver just recovers from it. We believe that this can give us a foothold in getting our cells in. The other thing is we already have pediatric dosing of acetaminophen. Acetaminophen causes pericentral necrosis of liver endothelium. It's actually very, very close in target effect to the monocrotaline. So since we already know the drug testing designs, and this is something that is given to a 1-year-old, I gave it to mine. So this is something that we could use to get cells to engraft a little bit better. But what cells are we going to put in? Remember, mine expressed GFP. That's not good enough, right? Also, my cells will be rejected as currently designed by everybody in the room. So we have to go to something called a universal cell. Universal cells are a cell type population that theoretically can go into everybody in the room because the immune system can't find them. One of the design variants is from Andras Naja's lab. These are immunologically cloaked cells. You'll see 8 different transgenes here that block all kinds of different immune cells, whether it's NK, T, macrophage, monocyte, dendritic cells, everything's being blockaded. So this means that the immune system can't find it, can't kill it. So we've made super cancer. Great. So that's a huge problem, right? Because now, what if that cell went awry, started proliferating like crazy? Now I've just made the most unsafe clinical trial ever. So now we have to introduce something that the Najee lab spent a lot of time building. It's called HSV-TK. Basically what it is a suicide cassette, but they didn't just put it in the cells, they linked it to the proliferation cascade that's required for cells to grow. So a growing cell is the only one that expresses this sensitizing drug. We can give them ganciclovir, the proliferating cells die, our graft does not. Really important design characteristics. So in an eventual patient, we give the cells to them, we give them a safe way of getting the cells in. If something goes awry, although we would have tested this, of course, we can give a drug that will kill the proliferating cells and leave the graft behind. We cannot kill the entire graft. If we did that, the patient's dead in a few minutes because all of their vasculature and their liver is denuded. So it would clot instantaneously. That's not acceptable. But a few cells dying, we believe to be safe. That said, what do I know? This has to be tested. This is very fanciful. Okay. Thank you very much for your guys' time. I want to, of course, thank the Gage lab that continues this research that was started in the Keller lab, where my postdoc was. We've had a lot of really important collaborators. Antonio Florenzi was really critical in this whole process, and a lot of core facilities. They've got a really strong animal facility that's made all this stuff possible over the many different spots. With that, I'd be happy to take any questions. If anybody's interested in trying to answer some of these questions with us, please give me a call.
There'll be lots of left there. So open it up to trainees first. Go ahead.
Why is this therapy targeted PM patients? Is the something that's a little bit
Would you mind repeating that for the audience?
Yeah, so for the audience online, so the question was, why does this therapy have to be targeted to that 1-year-old or the really early timelines in life? If we think about a risk management strategy, you'll get more benefit the longer you're able to use the cell. So an early timeline maximizes the benefit of the patient's lifetime. It increases significantly the risk to a lot of things that that patient might have. If there was anything that might change fertility, it's a bigger problem, right? So you do have to change and balance these things. To be honest, as a basic scientist, I shouldn't be and won't be making that decision in any reality. The other thing is a cell therapist. A small human needs less cells. So it makes my manufacturing a lot easier. A little bit of practicality, a lot of don't worry, I don't make that decision. Good clinicians make the decision. And the hematologist in the room will be deciding that stuff much more than anything else. Yeah.
In the long term, like after theoretical patients with T cells, like how would you ensure that the I guess native or not necessarily wild type, or I guess the unhealthy genetically or hepatic vasculature doesn't just regenerate and overtake the cells that because or if you're inducing some sort of injury to the cells and graft initially, how do you know that they won't just be eventually outcompeted as the starts to heal?
Yeah. So those are great questions. The question was, how do we know or control that the host endothelium doesn't just kick out our exogenous endothelium? So empirically, in the mouse, it didn't happen. We're still trying to understand why, to be honest. Monocrotaline is a bit of a dirty drug. We don't really know what it's doing to some extent. It's well studied, but in the liver, it's not actually well studied. It's really studied much more in the lungs, causing pulmonary hypertension. So there's a lot of good questions to that we hope to just skip, to be blunt. We want to move to systems that are not using monocrotaline. So we want to study it in ischemia reperfusion injury, in maybe the acetaminophen or angiopoietin, some other system that we want to get cells to engraft, and then it will be a really much more important question. In the mice, doesn't seem to happen. We can even do secondary transplants, and the cells are super fit and survive a second transplant and take over and fight the mouse endothelium again. But what if it's only a monocrotaline universe? Then it's basically clinically irrelevant. We'd have to test it in a better model.
Great talk. I have a couple of questions if I can. The first is that there is sinusoidal endothelium in the spleen as well. Could you comment on whether the cells that line that vasculature is similar to what you see in the liver? I mean, you do the transplantation. Is some of the repopulation in the spleen as well as in the liver?
That's a good question. So the first question is there differences between spleen and liver vasculature, endothelium? And the second question is, do we see engraftment in the spleen? Do the second one first. So we do see engraftment in the spleen. We have found to get engraftment in an animal in any location, you need a conditioning regimen. You need something that damages that spot. In the liver, we're using monocrotaline. In the spleen, comedically, we're wiggling a needle. So that causes some cells to engraft. We studied them very carefully. There's very small levels of cells, maybe 5 to 10,000 cells in each spleen, but when recovered, they don't express factor VIII. So I was really surprised about that. The factor VIII is driven as a gene product in the liver. That's all we really know from the human side. When we look at transcriptomic data, there are a lot of differences between spleen and liver, and factor VIII is much, much, much higher in the liver than the spleen. The spleen there's whiffs of factor VIII, like there is in a lot of organs. Nothing like lymph node. Lymph node is much higher. So I think that might answer your question. We don't really know a ton about the differences. I believe that the sinusoidal endothelium that sits in the spleen is now termed littoral endothelial cells, and they've got some special properties and really specific gene expression patterns, 20 or 30 or 40 or 50 genes or something like this that distinguish them from the liver. And I would say it's too new of a cell type to know if it matters.
One other quick question. So this endothelial cell heterogeneity is just crazy. I mean, the specification. The results on your single cell analyses show no von Willebrand factor in these cells. I think.
Yeah.
So BWF is made preferentially, we know, in the venous circulation. It's venous vascular endothelium. And yet here, there's no VWF. They're making the partner factor VIII.
Yeah.
The heck is that?
So the question is, why do my L6 not make VWF? And what was their crazy life choices, more or less? We don't know, but the quandary is from the same venous endothelial cell that I transplanted in, although it would have been hard to see on that one plot, and I'll go back purely for entertainment purposes. That little tiny cluster that I can almost completely cover up with my mouse, that has the von Willebrand factor. And if you look carefully, it makes a little less factor eight, but really it's the presence of von Willebrand factor. We have no idea, but at least we have cells to explore why for the first time, and cells, because they come from the same pluripotent cell, derived population, will be reproducible every time you look at it. And that's how we get to sort of these molecular questions. But as to why LSECs don't make one milligram factor, factor eight's so big, maybe they got tired. Very lazy answer.
Thank you.
So I was fascinated by the idea that you can use the, I guess, the geography of where of the tissue
Yeah.
to promote the differentiation. Have you tinkered with, I'm assuming that at the front end, your in vitro culture with the appropriate factors has some role.
It does. Yeah.
So I'm guessing that you worked long and hard to come up with that particular protocol to do that. So in the course of doing that, did you find that these cells appear in other places in the body other than the liver?
So in our engraftment model. So the question is, do we get engraftment in other sources? Oh, yeah, in other organs. Really sorry, Peter, I deleted that slide, so I can't show you the data. But what the answer was is we should have gotten fantastic engraftment in the lung. Monocrotaline causes lung damage. There was even a conditioning regimen there, and we don't. We look long and hard. We do not get engraftment in the heart, nor the lung, nor the femur, except in one very rare case of whole body irradiation. And it has to be in neonates in the 2-day window. Things that we don't really understand. The remarkable thing is the engraftment is very organ specific. I would love to be able to study the differences between heart vasculature and lung vasculature and liver vasculature from the same seed source, and we have no idea how to do it. And it's just sort of an oddball constraint, and at this point, just an empirical failure. We haven't figured out the conditioning regimen. We hope to develop other things, and maybe this ischemia system will help us get into other organs, muscles, stuff like that.
Maybe this is a silly question, but since you can infuse factor eight, you obviously don't need to make it there. So why wouldn't you just encapsulate those cells and put them in some sort of other setting where in fact you wouldn't have to damage the liver and then try and repopulate those endothelial cells?
Right. So it's a good question. So for those in the audience, why not just put the cells elsewhere and have the factor eight be pumped, more or less, a biopump? So Sigilon Therapeutics tried that, and what they did is a cell population that was a cancer-like line. They could control its growth a little bit, wrapped it in alginate capsule and rammed it in the IP space, and the body's immune system freaked out and got angry at the alginate and actually wrapped everything up and stopped it. So that was a bad design, but what if we had a mini liver? Right? A mini liver elsewhere. That's a good design. Challenge is, and we're back to the age-old problem of what are the factors and the design elements to put in that micro device so that we get enough factor eight being produced. Remember, most of this talk was that it needs to be a liver-like environment to get factor eight. Now, it could be a lymph node. It could be some other locations. It's not the only spot. Extrahepatic sources are real, but we don't know the design criteria for a mini liver yet. So at this point, we're cheating and borrowing the liver with the risks that it comes with. But it's still a great idea to try to make a pump from elsewhere, because in a terrifying scenario, you could cut it out. Right. It gives a really good recovery strategy.
Yes, I know nothing about this translational medicine stuff, but is there a translational medicine process that's approved where the first step is to bring the patient next to them and then try and repopulate that organ? Is there
yeah. So bone marrow transplant, right? Those patients are, their risk benefit ratio of that cell therapy is therapy or die, right? Like they're in that sort of space. Now, hemophilia, patients with hemophilia are not in that space. They have a much different safety profile. So we have to work on our safety game as cell therapists first. That said, there's a lot of different conditions, myasthenia gravis, MS, that is currently being treated with bone marrow transplants in Ottawa. So these other conditions that have different risk profiles get there. And we're actually going to be eventually leveraging the many, many different cell therapy designs that they're coming towards clinic for different conditions and hoping that they can do their safety profiling for us, more or less. But you're right. Hemophilia patients should be the last on the list, and they are. They will be.
Eventually, when you apply this graphical system in human patients, do you envision it as a off-the-shelf with immune cloned cell? Is that the way to go, or is there any consideration? I don't know, patient-derived iPSC-derived cells or anything of the sort?
Great question. So the question for anybody still online is, should we be going IPS sourced, so an autologous approach, or the allograft sort of design? I personally, I spent a fair amount of time around labs that have worked with big companies, and there is no company that is willing to invest in personalized iPS cells. And that just really shreds the monetary game for this. So if we have to work up our universal cell technology to give an option for somebody, I think that's just the way to go, only because at least there's hope of building a cell therapy. That does not mean it's right, though. Remember that it would be better in theory to make iPS cells for everybody on the planet and have their own personal cell repository. Current estimates are about $500,000 per cell therapeutic. It worked great when the Riken Institute wanted to do the first RPE transplants. That patient is seeing better. The second one failed because the cells didn't work out right. So it's like the money game is really a challenge. And in this case, these cells that the Naji lab have developed, I'm not a big proponent of them. I'm just saying that they're one example. They're already sending these cells to other cell manufacturing systems. So we can leverage and drop that cost to maybe $100,000 a patient. One could dream, but I think that the economics are going to drive this more than we give it credit for right now.
Question about the stem cells.
Mhm.
Do you know whether all those changes that are being made will interfere with your mesoderm development? I mean, you made this big comment. So there's a lot happening in those cells, which looks good for them to be immune cloaked. But what about the development of the cells in other ways?
Right. So the question is that, does the genetic modifications of universal cells disrupt developmental biology, more or less? This version of cells and other hypoimmunogenic variants have made endothelium. So that means it steps through a bunch of steps. Now, have they made my venous endothelium responding to BMP and activin? No, that's part of a series of grants that we're still sending out, and we don't have answers to those questions. That's why we have to ask those questions. Thus far, we haven't failed at making endothelium from any line. These could be the first failure, though. We don't know.
Anthony, yeah, go ahead.
Is your lab thinking about making this into a protocol of problems so that other labs can look at insects?
So, make sure I understand the question. Are you asking, are we as a lab generating cells for other labs? Yes. So we already have. I've got a freezer of about 200 to 400 million cells, depending on what flavor you want. They're red, they're green, they're male, they're female. Call me, you get them. The whole point was we build cells and democratize it. Endothelium is very easy to make. Please think that you can make it on your own. You can, it's okay. But the economics of making the cells are better when they're done at scale. So if I'm the manufacturer of cells and then I ship them to you and you do your interesting question, you focus on your research question, I focus on cell manufacturing for you, and you don't have to do both. Doing both is the problem. That's where it gets really expensive. And some of these protocols do not scale down well. You can't just make 1 million cells. You kind of need to make 100 million.
I'm doing this and anything other than mouse, yeah. Something that lives a little longer than a couple of years.
Well, no. So what's really interesting is the engraftment designs. Xenograft engraftment is a beast, and I choose the word entertainingly. So the xenograft in rats has different engraftment efficiencies. So we can't even go to the matched genetic rat. We get different engraftment. So we stick to our mice for our research space, knowing that eventually when we go to like a canine model or a different system or whatever, then we're going to have to make so many big jumps, then we will match to the target animal or patient eventually, if that makes sense.
Have you had another question?
Thank you.
Out of HS.
Yeah.
I know Andras has taught me